Hello friends, welcome back to my channel. In this video, I am going to give you 50 important MCQs. They are useful for your JL preparation, DL preparation, AP set, NET, PG set, NEET, MSET examinations. Okay, so let's see the questions one by one. First question is the most commonly used bacterial biopesticide is. Options are Escherichia coli, Salmonella typhimurium, Bacillus thuringiensis, Streptomyces griseus. For this, the correct answer is Bacillus thuringiensis, which is used as a bacterial biopesticide. Let's see the next question. Here, two lists are given. You have to match them with a correct answer, with a appropriate answer. Let's see the two lists first. First list consisting transgenic variety of crops. Second list consisting trait for which gene has been incorporated. Let's see the first list. Seedling maize, rainbow papaya, flower sour tomato. So second list consisting herbicide resistance, virus resistance, altered ripening. So I'm going to match the list 1 with list 2. So seedling maize, which is a herbicide resistant maize variety. Rainbow papaya, which is a virus resistant papaya variety. So flower sour tomato is produced for that to alter the ripening period. Okay, so that's why the correct answer is first one is the correct answer. Let's see the next question. The term biotechnology was first used by. Options are Colrater, Bullock, Carl Ericke, Waxman. For this, the correct answer is Carl Ericke is the correct answer. Let's see the next question. Which of the following is classified as a viral biopesticide? Options are Agrobacterium tumefaciens, nuclear polyhedrosis virus, Pseudomonas fluorescens, Rhizobium leguminosarum. For this, the correct answer is nuclear polyhedrosis virus, so which is a viral biopesticide. Let's see the next question. Nuclear polyhedrosis virus is a subgroup of. Options are retrovirus, adenovirus, baculovirus, poxvirus. So which is a baculovirus. Let's see the next question. A transgenic rice variety called golden rice was engineered by inserting genes from which of the following? Options are carrot and bacterium, daffodil and fungus, saffron and fungus, carrot and fungus. For this, the correct answer is carrot and bacterium. Let's see the next question. Retinone, a useful insecticide which is obtained from the roots of the which one of the following? Options are Loncocarpus utilis, Adathoda vasica, Humilis lupulus, Aloe barbadens. The correct answer is Loncocarpus utilis. Let's see the next question. Sensitive biological detectors are. Options are proteins, plasmids, bioherbicides, DNA probes. For this, the correct answer is DNA probes. Let's see the next question. Which one of the following is a non-symbiotic biofertilizer? Options are Acetobacter, Azotobacter, Frankia, Nostoc. Azotobacter is a non-symbiotic biofertilizer. Let's see the next question. Azalla is used as a biofertilizer because. Options are it multiplies rapidly and the decomposed biomass is a good source of organic nutrients. It contains rhizobia in root nodules. It lives in symbiotic association with blue-green alga. It contains mycorrhiza that provide phosphorus and potassium to the crop. Let's see. For this, the correct answer is azolla is used as a biofertilizer. Why? Because it lives in symbiotic association with blue-green algae. Let's see the next question. Which antibiotic was synthesized from a fungus during Second World War? Options are penicillin, streptomycin, oxytetracycline, erythromycin. For this, the correct answer is penicillin. Let's see the next question. So, bacteria are given. Among them, we have to pick which of the above synthesizes the substances toxic to insect. Okay. So, three bacteria are Bacillus thuringiensis, Bacillus papillae, Bacillus moriti. For this, the correct answer is first and second bacteria. They are used as a bioinsecticides. Why? Because they produce toxic substances. They are toxic to insects. Let's see the next question. Which organisms mostly produce interferons? Options are vertebrates, invertebrates, bacteria, fungi. For this, the correct answer is vertebrates is the correct answer. Let's see the next question. The genes CMPS, they are linked. Crossover map distances as determined by 2-point test crosses are P to C is equal to 7, S to M is equal to 10, C to M is equal to 8, S to C is equal to 2, and P to S is equal to 5. So, the relative positions of these four linked loci are, so by calculating these distances, you have to tell the positions of the four genes. Okay, so the positions given as PSCM, SCPM, PC, SM, SCMP. For this, the correct sequence, correct order is SCPM is the correct order. Let's see the next question. DNA probes are, options are isolated single radio labeled DNA strands used to detect the presence of the complementary strands, isolated double radio labeled DNA strands used to detect the presence of a particular protein, a sequence of DNA complementary to the functional mRNA, a small DNA that increases the rate of one transcription of a gene found associated with it. For this, the correct answer is DNA probes are isolated single radio labeled DNA strands used to detect the presence of complementary strands. Let's see the next question. In Agrobacterium tumefaciens, disarmed plastids differ from wild plasmids by not having them. Options are Vir regions of Ti plasmid, inducers of the Vir operans, tumor causing genes, and tDNA. Requirement for acetylserine gene. For this, the correct answer is tumor causing genes and tDNA. Let's see the next question. Example for a transgenic plant. Options are IR8 of rice, golden rice, Posamoti of basra, TMV tree of groundnut. For this, the correct answer is golden rice is a transgenic plant. Let's see the next question. Consider the following. So here also you have to give the correct statements. Insulin, human growth hormone, interferons, hepatitis B, surface antigens. Among them, which of the above are generated from genetically engineered microbes? For this, options are given 1 only, 1, 2, 3 only, 3, 4 only, 1, 2, 3, 4 only. For this, insulin, human growth hormones, interferons, hepatitis B, they are all generated by genetic engineering process only. Let's see the next question. Bacterial biopesticide is, options are Bacillus anthracis, Bacillus polymixa, Clostridium botulinum, Bacillus thuringiensis. So already told you that. So Bacillus thuringiensis is used as a biopesticide. Okay, let's see the next question. To impart viral resistance, plants can be genetically modified with which of the following genes? Options are chrygenes, chitinase gene, lectins, viral coat protein. For this, the correct answer is viral resistance can be given by using viral coat proteins. Let's see the next question. Tapai is a variety of this crop plant. Options are sorghum, wheat, rice, basra. Tapai is a rice variety. Let's see the next question. Which one of the following enzymes is added in many detergents that helps to remove proteinaceous strains from clothes? Options are alkylase, xylanase, peroxidase, catalase. For this, the correct answer is alkylase, which is used to help remove proteinaceous strains from the cloths. Let's see the next question. Golden rice is produced. Options are by introducing three genes for the production of vitamin A in Tapai variety of rice, by crossing IR8 variety with Posa variety of rice, by introducing genes from the production of high yield in Anamika variety of rice, by causing mutations in Basra variety of rice. Biraz variety of rice. For this, the correct answer is, so golden rice is produced by introducing three genes from the production of vitamin A for the production of vitamin A, tape variety of rice. Okay. Let's see the next question. Biosensors are used in. Options are in vitro diagnosis, detection of microbial contaminants in the food, monitoring pollutants in the abiotic component of the environment. So these statements, from these statements, you have to pick the correct statements. So for this, the correct answer is 1, 2, 3. Three statements are correct statements. Let's see the next question. Name the process of using living organisms to remove contaminants, pollutants, and unwanted substances from the soil or the water. For this, the options are bioinformatics, proteomics, bioremediation, genomics. So, bioremediation is a process through which contaminants can be removed. Let's see the next question. Which one of the following is the method of gene transfer based on the use of short electrical impulses of high field strength? Options are ultrasonication, microprojectile, electroporation, microinjection. Electroporation is a biological gene transfer method. Okay, let's see the next question. Baculovirus infects. Options are arthropoda, bacteria, protozoans, blue-green alga. Arthropods. Baculovirus infects arthropods. Let's see the next question. Consider the following statements. So, plants developed through suspension cultures generally exhibit genetic variations. Plants generated via organogenesis from callus cultures generally do not exhibit genetic variations. Which of the statements are given correct? So among them you have to pick the correct statements. Only first statement is the correct one. Let's see the next question. What is called the use of microorganisms for all biocontrol agents? Options are bioremediation, biopesticides, biotransformation, biofertilizers. So biopesticides is the correct answer. Let's see the next question. Shotgun technique is employed in. Options are stitching of the gene, isolation of the gene, sequencing of DNA, insertion of genomic clones into a vector. For this, the correct answer is insertion of genomic clones into a vector. Let's see the next question. Which of the following consists of both types of nucleic acids? Options are Bacillus thuringiensis genes, nuclear polyhedrosis virus, cytoplasmic polyhedrosis virus, tobacco necrosis virus. So, nuclear polyhedrosis virus consisting both types of nucleic acids. Let's see the next question. The plasmid were derived from E. coli. Col E1 is. Options are YIP, PYAC, PBR322, TI plasmid. So PBR322 is developed from E. coli. Let's see the next question. The biopesticide CPV is a. Options are bacterium, virus, fungus, nematode. So which is a virus? Let's see the next question. With reference to the designing of vectors based on the RI plasmids, consider the following statements. So they are disarmed by removing the genes causing the disease. They carry sites for the insertion of gene of interest. They carry selectable markers. So among these three statements, you have to pick the correct statements. So 1, 2, 3 statements are correct. Let's see the next question. What type of molecule or interferons? Options are lipids, nucleic acids, polysaccharides, glycoproteins. Glycoproteins are the correct answer. Let's see the next question. The shuttle vector is defined as. Let's see the options. A plasmid capable of replicating in one organism, a plasmid capable of accepting one large DNA inserts, a plasmid capable of replicating in cells, a plasmid capable of replicating in two different organisms. For this, the correct answer is a plasmid capable of replicating in two different organisms is called shuttle vector. Let's see the next question. PBR322, which is frequently used as a vector for cloning gene in E. coli is. Options are an original bacterial plasmid, a modified bacterial plasmid, a viral genome, a transposon. For this, the correct answer is a modified bacterial plasmid is the correct answer. Let's see the next question. The primary role of interferons is to options are facilitate viral replication within the host cells, stimulate the production of more viruses, protect uninfected cells from the viral attack, directly destroy virus infected cells. For this, the correct answer is they protect uninfected cells from the viral attack. Let's see the next question. The labeling of DNA probes, often with radioactively, is crucial for options are preventing the probe from degrading, enhancing the probe's ability to bind to its target, enabling the detection of a probe after it has hybridized to its complementary sequence, increasing the rate of hybridization. For this, the correct answer is enabling the detection of the probe after it has hybridized to its complementary sequence. Let's see the next question. Baculoviruses are primarily known to infect. Options are vertebrates, plants, arthropods, mainly insects, fungi. Yes, baculoviruses, they infect arthropods, mainly insects. Let's see the next question. DNA probes are valuable tools as highly sensitive to. Options are antibiotics, biological detectors, restriction enzymes, polymerase enzymes. So, the correct answer is biological detectors. Let's see the next question. The primary function of a DNA probe is to options are repair damaged DNA within a cell, introduce new genetic materials into an organism, detect the presence of specific complementary DNA sequences, catalyze the replication of DNA molecules. For this, the correct answer is detect the presence of specific complementary DNA sequences is the correct answer. Let's see the next question. Golden rice is a transgenic variety of rice that has been genetically engineered to produce. Options are increased levels of iron, higher protein content, beta carotene, a precursor of vitamin A, resistance to common rice pests. Yes, so golden rice is produced that consists of beta carotene, which is a precursor for vitamin A. Let's see the next question. Which one of the following plasmid vectors was constructed by combining centromere, telomere, and autonomously replicating sequence? Options are PBR 322, Cosmid, yeast artificial chromosome, nothing but yak, bacterial artificial chromosome, nothing but pack. For this, the correct answer is yeast artificial chromosome. Yak is the correct answer. Let's see the next question. How many genes were introduced to the tope variety of rice to create golden rice? Options are 1, 2, 3, 4. For this, the correct answer is 3 genes have been inserted into the golden rice. Let's see the next question. Which one of the following increases the frequency of agrobacterium mediated genetic transformation? Options are ascorbic acid, acetosyringone, aflatoxin, abscisic acid. For this, the correct answer is acetosyringone is the correct answer. Let's see the next question. Cytoplasmic polyhedrosis virus is another example of. Options are bacterial insecticide, fungal biopesticide, viral biopesticide, plant derived pesticide. So for this, the correct answer is viral biopesticide. Let's see the next question. The most commonly used method of choice for gene transfer using immature embryos as explants is. Options are microprojectile, electroporation, liposome mediated, chemically stimulated. For this, the correct answer is microprojectile method. Let's see the next question. Both nuclear polyhedrosis virus and cytoplasmic polyhedrosis virus belong to which category of biopesticides? Options are bacterial, fungal, viral, protozoan. So they are viral biopesticides. Let's see the next question. Consider the following statements. Agrobacterium tumefaciens is a soil bacterium. Agrobacterium tumefaciens was the first vector used for the introduction of foreign DNA into the plant cells. Among them, you have to pick the correct statements. Both 1 and 2 options are correct. So, okay students, if you like this video, so don't forget to subscribe my channel, give a like to this video also. Thank you.